So the ailment known as sickle cell anemia, so the sickling of red blood cells, is kind of your hallmark of the disease. That's due to a single mutation in the protein hemoglobin. Now, a glutamic acid is mutated, or is it's not mutated as if it spontaneously happens. That glutamic acid is converted to a valine, and that's found in the beta chain of hemoglobin. So what that means is, let's think about this in terms of amino acids. Glutamic acid, well, that's a residue for, well, you're going to have a carboxylic acid. And so it's going to have a negative charge to it. Valine, on the other hand, that's a hydrocarbon. So it's just a bunch of carbons and hydrogens on it. In which case, the interactions that can exist between glutamic acid and anything else, compared to valine and anything else, well, they're going to be changed drastically. This new valine side chain can bind to a different hemoglobin molecule to form a strand similar to the amyloidogenic proteins that we discussed in chapter 4. So there's that sort of aggregation. Now, what this results in is the sickling of red blood cells. So untreated homozygous individuals generally die in childhood. Heterozygous individuals exhibit a resistance to malaria. So it's what that kind of what the mutation, or what that amino acid mutation results in, is something that looks like this. So what we have is hemoglobin A versus hemoglobin S. Okay, so our sickling hemoglobin, what it's going to do is it's going to basically find other hemoglobin molecules and create contacts between them. And those contacts will be created between a large number of hemoglobin molecules. And those hemoglobin proteins will continue to aggregate and aggregate and aggregate, first forming these strands, and then alignment and crystallization to form these fibers. And so that kind of continued aggregation will result in a grander representation, a pretty significant change in the overall structure of a cell. It will result in that sickling of the cell. So that sums up everything that we did regarding oxygen binding proteins. We talked about hemoglobin, myoglobin, those binding curves, and everything that makes a little bit, all of that, sorry. That sums up everything that we need to go over with respect to oxygen binding proteins. But this chapter is all about protein ligand interactions and protein function. So our second case study is antibody and antigen interactions, two different types of immune systems. So we have a cellular immune system, which targets its own cells that have been infected and also clears up virus particles and infecting bacteria. Our key players are our macrophages, killer T cells, and inflammatory cells, or Th cells. Our humoral, or our fluid immune system, targets extracellular pathogens, can also recognize foreign proteins, and makes soluble antibodies. So generally when we talk about a so-called immune response, that's kind of what we're talking about, antibodies. This keeps a memory of past infections, and our key players are B lymphocytes and helper T cells. So our cellular immune system has antibodies that bind to fragments displayed on the surfaces of invading cells. Phagocytes are specialized cells that eat invaders, and macrophages are large phagocytes that ingest bacteria that are targeted by antibodies. Okay, so we have like an Fc receptor, for instance, and that is going to detect a IgG-coated virus. That protein detects that IgG-coated virus, and this macrophage is basically going to engulf, through phagocytosis, this antibody-coated virus. Now, our humoral immune system. Vertebrates also fight infections with soluble antibodies that specifically bind to antigens. Antigens are substances that stimulate production of antibodies. So these are typically macromolecular in nature, and they're recognized as foreign by the immune system. So coat proteins of bacteria and viruses are good kind of triggers for the production, or good examples of antigens. Surface carbohydrates of cells and viruses are also good targets, or also good stimulators, or also good antigens. Antibodies are proteins that are produced by B cells and that specifically bind to antigens. Binding will mark the antigen for destruction or at least interfere with its function. So a given antibody will bind to a small region of that antigen, and one antigen can have actually multiple epitopes. So, I think that in the past 3 months or so, because of COVID-19, we've heard the term antigen antibodies almost nonstop. But this is kind of where when we're talking about immune responses, and for those of you who have taken any sort of immunology classes, you've heard this well before. So an antibody has 2 heavy chains and 2 light chains. The light chains, one is constant and one has a variable domain. So the heavy chains consist of 3 constant and 1 variable domain. So these are highlighted as constant and variable. So 1 constant, 1 variable. So when we look at this diagram over on the right-hand side, we need to look at our legend to see what are the actual things that we're looking at. First of all, let's go ahead and highlight this guy right here. We've got our antigen binding site, then we have a variable light chain, a constant light chain, then on the opposite side of that we have a variable heavy chain and a constant heavy chain. Opposite of that, we've got basically the mirrored entity. We've got a heavy chain, a heavy constant chain and a domain, and a heavy variable domain, a light variable, and a light constant. And then down here, the remaining portion of this FC, we've got our constant chains, and they are all heavy, or our constant domains, and they are all heavy chains. So what we're looking for is, or what an antibody is looking for, is something that is going to basically be somewhat variable because it's got that variable heavy and light domain, or sorry, it's got those variable domains on a heavy chain and a light chain, and those variations are going to enable detection of new antigens. So what an antibody kind of looks like from I guess the ribbon structure of it would be something like this, where we've got our disulfide bonds right here that connect our individual proteins, and then here would be a bound antigen. In this case, it's a bound carbohydrate.